All right. So where we more or less left off talking in class, and I kind of want to circle back to this, is this concept that, uh, um, again, on the concept of translation and translation elongation. I'm going to set my pen here. And so again, as the mRNA is read through by the ribosome, and again, it's the ribosome moving over the portion of the mRNA and reading the mRNA in the amino ACL site to link a codon with an anticodon, and this being charged with an amino acid. This allows for the ribosome then to move, and as every subsequent tRNA binds to their anticodon, there's an ester bond being created in place of or along the amino acid or the amine group in the backbone, as seen here. So you see a connection between the amine group and the central carbon. And this allows for the R groups or the defining aspect of the chemical, structural, and polar attributes of each amino acid to to be out in space. And this is what eventually will allow for the various kinds of folding associated with amino acid interfaces. So as you mentioned before, right, there are certain amino acids that I'm going to ask you to remember for the test and that you should know just as part of understanding genetics as a whole. And that is the start AUG. And then, as I mentioned before, you have GUG, which can also be seen in a lot of prokaryotes, which codes for valine, and UAA, UAG, and UGG, all of which code for stop codons. Now, it's important to note that a great many different coding sequences, great many different codons, do sometimes code for a couple amino acids. Some of them code, or sorry, a couple might code for a single amino acid, or many might also code for a single amino acid. And then there are a few kind of rare exceptions, such as tryptophan, is seen here. Again, methionine and the stop codons, all of which code for a different release factor, which terminates translation. So elongation along the A and P sites of the growing polypeptide occurs, and when the peptide is fully formed, and the amino ACL site comes across a stop codon. When we see a stop codon, this stop codon is what is recognized by a molecule known as release factor. So, release factor binds into the A site. So, release factor 1 and 2 first binds, and this recruits release factor 3. Release factor 3 binds to GDP, which is then phosphorylated by the inorganic phosphate released by the hydrolysis of GTP. So this is where that uh, you know the emergence of succinyl CoA from succinate, right? So you remember succinyl CoA uh, to so succinyl CoA to succinate yields GTP. And as we said, GTP has a number of different functions, but as I mentioned before, GTP is really heavily involved in translation in amino acid um, polymerization during translation. This is one of the few different places where GTP plays an important role. So GTP is hydrolyzed, releases a phosphate, and this allows for the creation of energy, which releases release factor 1 and 2 and has release factor 3 still in tow, but removes it from the amino acyl site. So removing release factor 3 with the energy from the hydrolysis of GTP allows for the binding of GTP in elongation factor and release factor RF being bound. And when this complex, elongation factor G and RRF factor bind to the amino acyl site, this stalls the ribosome, and this ribosomal stalling causes the ribosome to shift without moving the small subunit. When this happens, the small subunit and the large subunit dissociate from the mRNA, and eventually the and eventually the small subunit is subsequently released when the last tRNA has been removed. And when the last tRNA has been removed, the small subunit releases, and the mRNA is subsequently destroyed as being a single stranded, unprotected nucleic acid entity. So it's important to note that, in the first two kind of binding of release factors, the first three release factors one and two and three, are really important for generating the energy required to release the polypeptide from the ribosome. When the polypeptide is released from the ribosome, two things have to happen. The first has to be the removal or the dissociation of the ribosome so it can be reused on another piece of RNA. But subsequently, the same thing with the amino acid that's released. The nascent polypeptide then can follow the steps of of folding that we talked about. So nascent polypeptide, primary structure, beta pleated sheet and alpha helix, and secondary structure, tertiary structure, with three-dimensional folding of helices and beta pleated sheets on top of one another, and then eventually quaternary binding in what we consider to be the uh, active or potentially activated or could be activated form of the protein. Now, it is at that level that we consider the phenotypic expression of cells and the phenotypes that they exhibit inside different circumstances, inside different environments. But as we mentioned before, phenotype is not only about the proteins that we produce, but it's not just about, you know, what proteins and what amino acid changes and or allelism we have in certain proteins, but like because that can affect function in overall phenotypic expression of the particular protein. But one of the biggest driving factors of how a protein will act inside of a cell, as far as its functionality, its has to be how much protein you have um, available to you. Now, as I mentioned before, the most common form, well over 90% of regulation of most genes is at the level of transcription. And again, this is because most chromatin in eukaryotes and or genes inside of prokaryotes are, yes, they do have some leaky expression off of their promoter, but they are typically controlled at the transcriptional level because it takes a great amount of energy to not only produce the nucleotides required for transcription and translation, but also to form the ester bonds to make a polypeptide, and then the energy in order to fold the protein, which is predominantly spontaneous because it's folded in the environment which it was born. But some of them do require chaperones and or are going to be utilized for energy consuming processes like catabolism and anabolism. So, because of all of these reasons and more that we haven't discussed, right, the level of transcription is the furthest most control up the chain of control that you can have in both prokaryotes and eukaryotes. Um, the next level of regulation that prokaryotes exhibit is translational control, which is how fast the mRNA is read through by the ribosome, and then post-translational control, which has to do with the fully formed protein and whether or not it's activated or inactivated off the ribosome, and in both of those cases, how frequently is an inactive form. So if it comes off the ribosome as an active protein, is there allosteric inhibition to regulate its activity? And if it comes off as inactive, the ability to have proteins at the ready and then subsequently activate them when they are required, especially important for critically important proteins. So just going off of these are the three major levels where prokaryotes and eukaryotes um, have conserved levels of regulation of genes. For eukaryotes, and especially since some eukaryotes are microbes, it's important to note that all eukaryotic microbes, such as again fungi, um, eukaryotic viruses, and protists, they utilize a great many of other things, including the fact that they have transcription that occurs inside the nucleus and translation that has to be, again, mRNA that has to be um, migrated from the nucleus to the cytoplasm. These include chromatin remodeling, which is because, again, for eukaryotes, right, all of the DNA is bound by histones, and it's in these open or free DNA where they're not bound by translation, not bound by histones, that transcription factors can cause the chromatin to shift, right? It can move and mobilize the histones, and by shifting and mobilizing the histones, there is subsequent um, rollout of more DNA. So if you had something like this, and you had DNA that's wound like so, when you add a transcription factor to the open DNA, you end up getting a little bit more DNA accessible and then a little bit more DNA accessible until you're able to read through the whole transcriptional unit from the 5' UTR to the 3' UTR. Um, there is post-transcriptional um, control. Post-transcriptional control typically is in the form of processing the pre-mRNA. So this is the removing of introns um, after transcription. So after transcription, you have to remove the introns to have solely an mRNA transcript which contains exons. Um, some evolutionary perspectives on RNA splicing have led to something called alternative splicing, where you get exons and introns and exons, and instead of getting um, instead of getting all exons, you might have an mRNA that looks like this, where an intron or a piece of an intron has been left in. And this is something called alternative splicing, meaning that the splicing event did not occur correctly. And then this can change, obviously, the amino acid composition of the gene. And sometimes this is worked out in favorable situations, such as it's widely believed that alternative splicing is how a great many of the immune signaling molecules and receptors work in the human body, whereas sometimes it's very detrimental, like the inclusion of an intron can often lead to genetic disease. So there is a fine line between true RNA splicing and alternative splicing. Um, the gap between post-transcriptional and translational control can also be secondary expression of regulatory mRNAs, so usually called miRNAs. So or silencing RNAs, um, or RNAi or RNA interference, and these are typically non-coding. These are non-coding RNAs, and basically how the non-coding RNAs work is that they, you might have more. Um, how these work is that the RNA molecule exists as an RNA molecule and it's bound by a ribosome. However, if the RNA now binds to a second RNA molecule, this duplex, double-stranded RNA species cannot bind to the ribosome either mechanically, and also double-stranded RNA is typically the target of again, because it's an irregular complex nucleic acid species, it can be targeted by both the CRISPR system in prokaryotes and by the Drosha complex, the slicer dicer complex inside of eukaryotes. So, because the vast majority of expression occurs at the level of transcription, important to note things that we've already talked about are, constitutively expressed genes do not fall into the category of being regulated because they are typically transcribed and translated under all conditions. And so this means that they are essential to the function of the cell. So the vast majority of genes, even genes important for energetics, thermodynamics, and physiology of cells, the vast majority of those can also be regulated. So there's a very small populace of genes which are constitutively expressed. And these typically result in genes that the cell cannot live without, like absolutely 100% cannot live without. So we call these housekeeping genes, um, because they are always expressed at the same level. And the cool thing about housekeeping genes is not only because they play an important role in like everyday cell function, they also serve as an important role or a certain control if you're looking at expression differences in genes and in protein synthesis. So if you want to know if your gene is upregulated or downregulated, you can always compare it to a very steady state control by constitutively expressed genes. So it's an important finding for us experimentally because we can utilize these in Western blots, Northern blots, different types of expression-based assays such as gene reporters or RNA sequencing, et cetera, so on and so forth. But the vast majority, including a lot of genes that are essential for cellular physiology and function, are regulatable, regulated genes. And regulated genes can be either induced or upregulated in expression. So they are transcribed and translated at higher levels in response to an inducing factor. And this factor typically is a transcription factor that has something to do with a cellular sensor. So typically how regulation works is that there is a sensor that regulates the genes involved in that biogenesis or biosynthetic pathway or biochemical pathway. When that sensor is triggered for an increase in substrate or an increase in some type of signal, it triggers the activation of a transcription factor. And again, the transcription factors help aid the access to the promoter for RNA polymerase. RNA poly. And and so if you can turn genes up and and can overexpress them or express them at a higher level because of a change of environment, when that environment is no longer inducible or conducive to expression of that gene energetically, you need a way to turn it down, or else you're going to burn out of energy. And this is where we get the concept of repressible genes, so genes that can be repressed or turned down. So anything that can be induced can be repressed. Anything that can be repressed can be induced. Now, again, typically speaking, when we talk about eukaryotes and prokaryotes, eukaryotes have these vast gene barren distances in their genome. And this is because there is a significant amount of complexity in gene regulatory regions. And again, we talked about these in class, the concept that there are DNA sequences. So there are DNA sequences which can bind to certain proteins, and those protein DNA interfaces open up the promoter for RNA polymerase to bind. Right. And so so what we see here is, again, this dynamic between the ability to go from sensor to regulating some aspect of the physiology of a cell in order to change some aspect of their transcription translation processes. Now, in eukaryotes, there's a number of different ways this happens, including interactions with chromatin, the fact that we have multiple chromosomes, the exons, introns, and we have these vast 5' UTR regions where there's nothing going on, and then there's a gene. So this leads to DNA sequences, which can be enhancers. So we have enhancers, and these are elements inside the DNA. They call enhancer elements that bind to transcription factors that are enhancers, and these increase transcription. And then there are insulators. There are insulators, and insulators prevent or downregulate um, expression. Now, typically how this happens is enhancers very simply enhance the ability for RNA polymerase to bind the promoter. Insulators work in kind of two different particular ways. Insulators can either work by closing off the promoter. So let's say here's the promoter. Insulators can either sit in or close to the promoter, or change the structure of the DNA around the promoter, which prevents RNA polymerase to bind, or it can actually sit just it can sit just downstream of the promoter, like so. And so if RNA polymerase binds here, RP, and this is an inhibitor or an insulator element, RNA polymerase is actually mechanically, physically blocked. In prokaryotes, when we have a regulatory molecule that exists, or regulatory sequence that exists between the promoter and the start of the first gene of the operon, we call this an operator. So the operator serves as a regulatory gene that regulates the ability for RNA polymerase to bind the promoter, and not only to bind the promoter, but also to read through the promoter and the transcriptional units in that locus. Now, some of these work again by mechanical functionality, and they just prevent RNA polymerase from reading through. Some of them will bind to RNA polymerase and activate. So operators in prokaryotes can be enhancers. They can also be insulatory, in that they can either block and prevent RNA polymerase from binding and or the ability to read through, or they can aid in the ability for RNA polymerase to bind. Now, typically speaking, enhancer function in cis occur upstream of the promoter because that helps access the promoter a little bit more efficiently. However, some of the operators can be positive and negative, depends on the operator and depends on its regulatory function. And to throw one more important wrinkle in this is the concept that if again you have a cell and there are sensors on the outside that are connected to the cell, and there are sensors on the inside, right? These are weighing, the importance of certain pathways to the cell all at once. So you get this oscillatory functionality in operators and in elements in cis, that sometimes you'll have so in a certain gene, right? Here's the promoter. In some cases, you might have enhancers enhancing transcription factors binding certain components, and then inhibitory operators or insulatory operators binding operators binding the operator sequence. And in this case, when you have both of these, one that's going to increase transcription, one that's going to decrease transcription, this now becomes a power of will. Depends on how much signal you're getting. If you're getting more positive signal, you get increased expression, because typically when these are bound, it'll knock off the insulator. Typically when the insulator is bound, it prevents the binding of enhancers. So it's this cat and mouse game, for this back and forth of when genes are being upregulated or downregulated, much like we talked about with the flagellar expression. It's not an all or nothing situation. This is very much a dynamic process by which cells respond in real time to how the signals from their environment coordinate with the signals from inside the cell in order to drive gene expression. And again, this all has to do with the fact that typically if something is regulating the expression of genes on a singular operon, it's because this whole entire operon has a series of proteins which all are involved in the same biochemistry or physiology or response, or some type of other phenotypic response or activity of the cell. And so you're kind of coordinately regulating a great number of proteins to be activated all at once, instead of activating each individual protein in response to a phenotypic requirement of that cell, what you see in eukaryotes. So these sensors work in the same way that biochemical sensors work, in both positive feedback and negative feedback loops. The vast majority of these are negative feedback loops, because again, this is all about energy conservancy, especially when you're talking about prokaryotes. So the vast majority of these are negative, in that the output that you get in a system negatively feedback, not only in the enzymatic allosteric functionality, but also typically in the transcriptional allosteric inhibition of the production of the enzymes in certain biochemical pathways. And some, like in the case of quorum sensing, right? So quorum sensing is a good example of one of these in prokaryotes, where the system has an output, and then that output feeds back to generate more output from the system. We've already seen this before, so I won't spend a ton of time on this. But again, when you have a biosynthetic pathway, energy conservancy is the name of the game when you are a prokaryote, because again, your change of environment, change of circumstances happen so immediately fast, which is why, again, evolutionarily, the combination of transcription and translation, as we discussed kind of ad nauseum in this class, is essential, because it allows the bacteria to respond to immediate and sudden changes in their environment. And those changes do happen in far more frequently alternating existences than our environment. When we have a day where it's 63 degrees outside and sunny, the whole day exists like that. And we can walk over and get food and stuff like when we're hungry. But for prokaryotes, the availability of nutrients, the availability of food, the availability of correct environmental conditions can change in a second's notice. And so the ability to conserve energy by regulating genes that need to be turned on versus off is essential. So here again is the tryptophan negative feedback loop concept here, right? Tryptophan allosterically inhibits enzyme 1, but it also binds to the transcription factor, which binds to the promoter. So if we think about here would be the promoter, here would be a regulatory region over here. So it binds to a transcription factor, which blocks or inhibits not only the function of the enzymes that are already produced, but it inhibits the expression of the operon in totality. So you're not spending energy in the form of GTP for transcription and translation. and you're not spending extra ATP by the biosynthetic pathway of the function of enzymes. Now, transcription factors come in four different forms depending on how they work. Uh, One is a what we call a positive inducible transcription factor, where the activator exists, and in the absence of that signal, whatever that signal is, right? So if there's no signal, there's no transcription because there's no added RNA polymerization from binding to that element. However, when the inducer is added, when we add in the inducible signal, it induces the transcription factor to bind its cis element, and then this activates the ability for RNA polymerase to bind the promoter, and it increases availability of that promoter, and therefore increases transcription and translation. There's also negative inducible, which is where you have a repressor. The repressor sits and either prevents RNA polymerase from binding or prevents RNA polymerase from reading through. However, when there's an inducer introduced, when we're trying to activate transcription, um, the repressor or the operator will actually the repressor, the regulatory gene will bind the inducer. And again, this is all has to do with allosterics and substrate binding, right? The substrate for these transcription factors is the DNA sequence that they bind. When they are allosterically conformationally changed by binding to an inducer, they come off the DNA because they've changed their conformation so that they no longer recognize the DNA. And this allows for RNA polymerase to bind and inducing transcription. Now, repression can also occur in the same way. So we have positive inducible, negative inducible. We also have positive repression and negative repression. This pathway, right over here, looks a lot like this pathway here, where it's still bound, right? So this is bound to the cycle. The only difference is when it's instead of inducer, if it's a repressor, when we add in a signal, when we add in a signal that is repressible, it binds to the transcription factor and allows it to bind to the cis elements. Whereas, and just so we're keeping these nice and simple, let's change the color here. Whereas, this is so this would be how positive uh, so yes, this would be an example of positive repression, and then negative repression would be the exact opposite. So it would be the ability to take an activator off, right? So if you had an activator and you were able to take the activator off the insist element because of the addition of a molecule, then you would prevent transcription by the removal of that activator, which is the same thing as inhibition. So inhibition can occur either by let's see if I can okay, so inhibition can occur either through RNA polymerase, so blocking RNA polymerase, either binding or reading, which can either be done by the addition of a repressor or removal of an enhancer or an inducer. So again, this matters because the ability to activate and transcribe and translate genes, because of the relative location of genes and coding sequences inside of prokaryotes, are basically toppled on one another. It's really essential that bacteria can read in all three frames in both directions. So um, when we think about the fact that uh, each codon is three nucleotides. It has three nucleotides on one strand, it has three nucleotides on the other strand. And as long as RNA polymerase is still reading in the 3 to 5 direction, it means that each one of these can start or be the starter, um, um, can be a starter codon for any gene, as long as there's a promoter, as long as there's a promoter, right? As long as there's a Tata box and a Pribnow box, so -35, um, and a -10 element, as long as these two things exist, um, it doesn't matter where these exist in relationship to either one of these three nucleotides, as long as they eventually create a start AUG or GUG. So bacteria, unlike eukaryotes, actually read off of both the top and the bottom strand, and so they have 6 reading frames um, in their arsenal in order to um, regulate gene expression. So this is one really intricate kind of aspect to gene expression, in that when you have binding of an activator or a repressor, it can affect the gene that it's actually bound to. But because of the 6 reading frame issue, it can also change the transcription and translation of other genes that are around it. This is something called genetic polarity. So the concept that the structure and function of transcription, translation, and expression of a gene based on its sequence that can affect neighboring genes and their expression as well. And not for nothing, but again, it's really important to understand that this is definitely a not an all or nothing thing. So what this leads to is varying levels of gene expression. So if you look at gene expression over time, it wouldn't just be this. If it was being induced, gene expression looks something like this, where you're getting these highs and lows, but you're getting a gradual directionality in either direction. So this would be induction, so this would be induction greater than repression. But if repression is greater than induction, you get kind of the opposite, ups and downs. But again, the general trend line is approaching that particular thing. So this can affect things like uh, gene dose, so how much protein you have, and that can affect that can be a direct, uh, effect on phenotypes. And if that's a direct effect on phenotypes, then that changes how that gene functions inside that particular situation. And not for nothing, but all of this is complicated also, not only because of the ins and outs of the signaling, and that these genes are all subject to regulatory conditions that is associated with non-Mendelian version of inheritance, because this is not a diploid organism, right? These are haploid organisms. So that means that things like incomplete dominance or incomplete expression, or um, um, sorry, and the like, even though they're not diploid, if they have alleles or paralogs inside of their uh, inside of their genome, which a lot of them do, or genes that have functional redundancy, meaning different genes that have the same function, all of their ability to be regulated is heavily dependent on the signals that the cell gets based on the environment that they're in. And therefore, you get this kind of turn on, off, but eventually to a directionality to affect phenotypic change. Part of this is also because, again, RNA polymerization, just like in transcription, just like DNA uh, replication, is not just simply, binding of RNA polymerase. It's RNA polymerase and all of the factors associated with transcription. And if the promoter and the rest of the elements inside the promoter region are not available, or if there's been a frameshift mutation or change in repressor or inducer functionality, that's when you start getting changes associated with um, transcription levels and therefore translation levels. So just take a moment. So, so going back to the tryptophan example, typically tryptophan has a repressor that binds to its operator. When tryptophan is in excess, it binds to the repressor and maintains expression of the repressor on the operator, and this prevents RNA polymerase read through because in this case the operator exists downstream of the promoter. However, when tryptophan is lacking, when there's absence of tryptophan or not enough tryptophan to reach saturation, there's not enough tryptophan to bind to the repressor cannot bind the operator, and therefore RNA polymerase is able to bind the accessible promoter and read through the entire operon. So glucose has a really well expression, has a really well described expression regulatory sequence as far as sugar catabolism is concerned, because again, glucose is the molecule that everybody wants, but it might not be the molecule that everybody has. And this is typically not any more relevant than when you have high levels of AMP. So AMP, as we've discussed, a really important regulator of all of the catabolic and anabolic processes that come off of glucose metabolism. And because of this, the more AMP you have, the less functional your anabolic processes are off of the glycolytic and the TCA cycle to create lipids, nucleic acids, and amino acids. So typically speaking, when AMP is in high amounts, it can be converted to something called CAMP, and CAMP or CAMP is created by something called adenylate cyclase. So adenylate cyclase is a really critical enzyme that basically converts AMP to cyclic AMP. And cyclic AMP has a structure that can be bound by a great number of sensors and serves as an energetic response element inside of cells. And as a lot of you have pointed out to me, CAMP can bind to and affect phosphofructokinase regulation. It also helps the cell trigger its ability to recognize and catabolize different types of sugar bases to make them into glucose. One of the most well-defined of these is the lactose operon, which has enzymes that convert lactose back to glucose and in order to utilize for the glycolytic cycle. So how this is regulated is when glucose is absent and lactose is present, CAMP binds an enhancer site. So here we go. Here's an inducer site or an enhancer site, also known as the cap site. So cyclic AMP binds to the cyclic AMP transcription factor. This binds the cyclic AMP transcription factor site which allows for the attachment of RNA polymerase to the promoter. This dynamic is mechanically regulated. So this is or signaling regulated. It's also mechanically regulated whether or not the repressor of this operon sits on the operator. In the presence of lactose, the lactose binds to the repressor and therefore activates the genes that are important to convert lactose into glucose. If there's no lactose, you would still, if you had no glucose and no lactose, you would get this binding. However, you would get this binding. However, the repressor would sit on the operator, preventing transcription. So this is how this system works, in that when there is no glucose, so there's no glucose, and so when there's no glucose, the ability for CAMP to bind the cap site and then the cap to bind the cap site happens in basically all of the catabolism operons in any given bacteria. So whether they have an operon for lactose, fructose, sucrose, etc., all of those are going to bind, or they will all have response elements regulating the ability to bind CAMP. The inverse is true for things like the pentose phosphate pathway or the polymethylation pathway or the amino acid biosynthetic pathway, where CAMP binds cap and CAMP plus cap binds to repressors on operators and they prevent amino acid biosynthesis, the pentose phosphate pathway, and the polymethylation lipid pathway, the lipid biogenesis pathway. So when we have this, CAMP will positively induce sugar metabolism, but negatively or will repress biosynthesis of other macromolecules. And this makes sense because, again, the reason you have AMP is because you have lack of ATP. So in the absence of glucose and in the absence of any sugar that this binds, but this is the lactose operon, so we'll go with this as an example. In the absence of lactose, the lactose cannot bind the repressor. You still get induction of you still get binding of CAMP and cap. So RNA polymerase, this recruits RNA polymerase and the other transcription factors to the motor, but now it cannot. So it doesn't prevent binding, but it does prevent read through. So the RNA polymerase cannot read through the operator and therefore cannot transcribe the lac operon, because, again, evolutionarily speaking, why are you going to make enzymes for a biosynthetic process making lactose into glucose if you don't have lactose to begin with? However, when you have the absence of AMP, right? So when you have high levels of ATP, there's no AMP. If no AMP is bound, again, this goes back to the leakiness situation. RNA polymerase is going to bind because the promoter is relatively open, and there's no repressor because there's no lack of glucose, so no repressor on the operator. There's no AMP because of the lack of glucose. This allows for a low rate of transcription and translation of the lactose gene. When there is a high level of AMP, and so this is plus so this is sorry so this is plus glucose plus ATP, no lactose. This situation here is plus lactose. So when we have lactose but no ATP, which creates AMP, and then we have no glucose, which means no ATP, which then cyclizes to this. And so this, again, lactose will bind to the repressor because the cell is sensing lactose, and This takes the repressor off the operator, allowing for a significant increase in RNA synthesis and translation. Um, and so these kind of combine to regulate um, how much of how much and of what sugar catabolism genes had to be expressed at a given time in order to generate a increase in glucose stores to drive central metabolism. Now, speaking, the vast majority of genes, and if you're thinking to yourself, well, if I was to take, say, a type 3 secretion system and all the effector molecules that might come into a type 3 secretion system and the type 3 secretion system itself, it would be hard to imagine that those would exist in a vacuum, that one single operon could control the expression of all of those things. And for that matter, that there's only one or two regulators that regulate the expression of those genes. So typically speaking, there are there are things called direct regulators, and direct regulators are regulatory proteins that control the expression of a single operon. Um, and then we have things called universal or global transcription factors. And universal global transcription factors are what we call regulators that influence a number of different programs, not different genes on different operons. So, in some cases, we could have um, a gene set here, and then we could have a gene set here, and a gene set here, and all of these operons, if they are all regulated at the same time, if they're all upregulated in the same way and downregulated in the same way, we call these collection of genes a regulon. And a regulon basically is different locate genes that are in different locations on the chromosome that are coordinately regulated by the same set of signals. So they're not all in the same place, but they're all regulated by a series of transcription factors, which are respondent to a particular set of signals. Most regulons are regulated both by direct or single function uh, transcription factors, so transcription factors that regulate each individual promoter region of each of these uh, operons, so they can have their own individual ones. But if that's the case, each individual transcription factor has to be able to respond to the same signal in the same way, or they are regulated through a universal regulator. So one of the most well studied is the RPOS starvation response, in that the RPOS gene or the RPOS protein, when cells under extreme starvation responses, will activate a number of different operons, including antioxidant, uh, creating antioxidants, acid, and alkaline resistance. It will start to activate. It will start to repress anabolic processes. It will start to increase catabolic processes in order to try to overcome the starvation response and in order to survive any particular insult that might happen to the cell um, while it's under extreme amount of stress already because of the starvation. So regulons, and I'll just spell that here for you. So regulons are basically series of genes that are either coordinately regulated by their own individual transcription factors or regulated by a universal regulator or both, But the concept of a regulon in a universal transcription factor is that there are ways in transcription factors and gene loci operons which are coordinately regulated in order to perform a phenotypic function for the cell to respond to whatever the cell requires to respond to in that particular environment that the cell finds itself. Now, most of these sensors and ways that bacteria, especially, but also eukaryotes for that matter, can interface with the environment, is that there has to be there has to be a way to harmoniously respond to changes in the environment in both the nutrients and the signaling molecules and the environment itself, as well as the health of the cell physiologically inside of the cell. So it's a combination of of the ability to communicate with the environment, or extracellular communication and intracellular communication. Now, extracellular communication and intracellular communication depend predominantly on what we call relay systems. Now, there are a couple different kind of versions of this, in that sometimes extracellular signaling molecules can internalize themselves and become transcription factors. So basically, the receptor binds to a signaling molecule. The signaling molecule becomes its own transcription factor. Um, Another version is what you're seeing here, where you get binding of a signaling molecule. This causes a histidine or a tyrosine kinase to phosphorylate one of those residues, and then you get a phosphokinase relay system that basically signals down to its amplification or transducer protein, um, and eventually causes a change in either genetic or protein functionality. So these relay systems can influence gene expression um, by way of transcription, or it can influence phenotypic gene expression through uh, protein um, activation or inactivation. So it can either transfer these relay protein. You can relay the phosphates or other signaling molecules like methyl groups or acetyl groups onto proteins to activate them or turn them off. Um, but effectively, so we have individual transcription factor conversion of receptor molecules. There's also relay proteins, and there's also the lipophilic and the small uncharged lipid or lipophilic signaling molecule, which can bypass the membrane and might have an intracellular receptor, which can undergo either direct transcription factor functionality or be part of a relay system. The vast majority of these results is that, well, what this results in is either you get relays from one system that either activate a multitude of other systems, or can just relay information to regulate expression or function in a singular system. And one thing that you have to understand about this is that because of the oscillation between inhibition and activation and induction of most genes inside of genomes, both prokaryotic and eukaryotic, there's a bunch of different proteins that are involved in these signaling pathways, and some of them are amplifiers that basically ensure the signal reaches its final destination, whether that destination be change in transcription or a change in protein function. Uh, um, or this becomes a situation where you get alternative pathways which can be involved, or divergent pathways which can be involved, And these can often affect the central pathway, right? Usually these side pathways, let's say that there's an inhibitor to this pathway and you're trying to activate it. These side pathways can inhibit an inhibitory pathway, which helps again amplify that message. But maybe you're also getting, let's say, a gene is, again, let's say like the RPOS, the starvation signal, right? You're going to get both repression, right? You're going to get repression of the pentose phosphate pathway and the amino acid biosynthesis pathway. But starvation isn't only sensed by RPOS. You're also going to get other types of um, stress activating proteins. So you're going to, again, going back to the fact that you have this kind of increase or this kind of decrease where you have general trends of activation, general trends of repression. So this all comes from the more signal you get to activate, the more linear this line becomes, the more the more you get for repression, the more linear this line becomes. Or the kinetics change in that you get more asymptotic activation or repression of that gene. So overall, these relay systems are really important in how cells, especially prokaryotes, integrate both extracellular signaling messages and intracellular physiological signaling messages to the cell itself. The vast majority of these pathways, even though some of them do use other alternative methods such as acetyl groups or methyltransferases. The vast majority and the most well characterized of these are the phosphorylation cascades, also known as the phosphokinase relay systems. So, so the phosphokinase relay. And basically, how these work, and they're very energetically expensive in that they hydrolyze ATP to not only cause a change in conformation and activity of these proteins, but also free the added inorganic phosphate, which can change conformation to allow for ATP to be received by every subsequent protein. And eventually you get to what we call the active target, whether that be a transcription factor or a protein of interest, and then you get a subsequent cellular response. And again, this can either be by protein function, so either function or synthesis by way of transcription. And the ability to sense things inside of cells is vast, right? And it all depends on where you find those particular signaling molecules. So, for example, things like sporulation or chemotaxis and oxygen osmotic sensing, there is a great deal of different molecules, but they all basically have some type of initiating signaling apparatus. So there has to be a signal receiving domain, and that receiving domain depends on where you find the thing. So where you have water on the outside, or tonicity is sensed on the outside, oxygen tension is sensed on the outside, chemotaxis is sensed on the outside, and same thing with environmental conditions, which regulate sporulation, sensed on the outside. However, as we mentioned before, there are also intracellular receivers of nonpolar, uncharged aliphatic molecules, such as quorum sensing molecules, which can have both extracellular binding domains in two-component regulators and also monocomponent regulators or single component regulators that exist inside of the cell. So, we talked a lot about changes in transcription. This, one of the examples of cellular responses as it relates to signaling inside of cells, that's a really good one, is the flagellar signal transduction pathway. So, the transcription factor function changes expression in number of proteins available. But cellular responses can also be physical or physiological to the proteins themselves. And as I just mentioned, a lot of these are based on phosphorylation cascades, such as the kinase key A, which phosphorylates key Y. In this is the natural state in which this is the natural state in which um key Y can then bind to Fly M, and this leads to clockwise and tumbling motility inside of cells. Tumbling, sorry. Um, However, when there is activation of the two component regulator, right, there is um uh the ability to chemotactically sense different molecules, and depending on what they're sensing, this can change the interface between key W and key A. And when key A is autophosphorylated, it can also phosphorylate key B. And key B is a methyltransferase, which means that it's a methyl esterase, so it takes off methyl groups. So in the case where key A is not phosphorylated, because it's phosphorylating key Y, when the autophosphorylation doesn't occur, key R can transfer methyl groups to these molecules. And when key R methylates, when key R methylates the receptors, it turns off the phosphorylation event between key W and key A autophosphorylation. So this then prevents key Y phosphorylation, and the activity of key Z plus the absence of phosphorylation off of key A results in counterclockwise motility. Whereas when key B removes where key B removes the methyl group, these receptors are now not as receptive to their inducers, and therefore they allow for key A autophosphorylation and prevent or predominate the clockwise motility. So again, this is where you have, again, key B making methyl groups, key B making methyl groups allowing for the sensing of an attractant. And so key B, again, making the ability to sense an attractant, blocking phosphorylation of key A. This allows key Z to dephosphorylate the auto-populated key Y, which is phosphorylated, resulting in a run motility. But again, because key Y is so heavily over phosphorylated, you get run and tumble. Again, here is like this kind of function of repellent and attractant, right? You get run, tumble, tumble, and now as you start to enter into the green, you start to sense the attractant. You run, tumble, and then run, as you are again, and this would be, as you get closer to your repellent or your attractant, these would be plus methylation, and again, plus methylation, in order to become more sensitive to the ability to receive those attractants and repellent molecules. So just kind of finishing this off, the concept of signaling, there are a lot of different types of signals, right? Oxygen tension, water tension, basically everything and anything that a cell has to respond to in that particular environment that it lives in its environmental range is typically part of the signaling molecule network as far as the extracellular components are concerned. And then the intracellular components or the intracellular regulators are more have to do with physiological behavior and metabolism regulation. There is one sense of signaling that occurs inside of bacteria where it's kind of in between both the extracellular and intracellular functionality, and this is the concept of quorum sensing. The general concept of quorum sensing is exhibited up here, whereas the ability to form a quorum or having enough individuals to make a decision. As you have low cell density, you only produce small amounts of a particular molecule, but the more individuals you have, the more molecules you produce, which can induce group behaviors. And again, this is important because, again, you need a way to regulate individuals as a community that are not tethered together in a cell-to-cell dependent manner. So fungi, protists, they all have different mechanisms by which they can do this. Eukaryotes, especially multicellular eukaryotes, definitely can do this. But prokaryotes don't. They don't tether themselves to themselves other than maybe by protein interactions, but never by cytoplasmic interfaces. So the ability to get the group or the species to behave as one is an essential output in order to generate the ability for the species to survive. So typically group behaviors, especially associated with quorum sensing, are heavily involved in regulating everything from virulence and pathogenicity genes all the way down to even like group metabolism and biofilm structure and composition. Um, the general concept of quorum sensing is when you have a great amount of a signaling molecule, they can either bind to a sensor kinase, both either a two-component sensor kinase or an intracellular sensor kinase, because most of these, so AHL stands for acyl homoserine lactone. So acyl homoserine lactone, also known as autoinducer 1. And for gram positives, they use their AI1 system is something called a quorum sensing signaling peptide. So because these are peptides, they're charged, so they cannot just pass through the membrane. So whenever you have signals that have a charge on them, they cannot pass through the membrane, and therefore their receptor has to be a two-component regulator. For acyl homoserine lactones, there is no charge and they're relatively small and they're non-polar, so they can pass through the membrane, and their receptor is actually an intracellular receptor. And these systems are predominantly, as I mentioned before, are feed forward. These are one of the various examples of feed forward or feedback forward positive systems where the gene can bind to the promoter and it expresses the genes that's under the regulation of the quorum sensing regulator. But one of the promoters that is also the same, in this case, the Lux gene, or whatever gene regulates the expression of these particular molecules. So quorum sensing is kind of an intermediate signaling system compared to environmental and intracellular physiological respondent elements, because they are, as you might imagine, most bacteria live in complex communities. So they are an environmental regulator because it changes the dynamics of the signaling molecules from other microorganisms and the microorganisms in your species, but also play a huge role in the intracellular component of this because they have an intracellular regulatory response element that helps regulate the physiological response of those particular cells. So if cells are needing to act differently physiologically to survive a particular environmental condition, quorum sensing can help regulate the what's going on the outside to what's going on in the inside. a lot, and this is where the complexity of signaling goes, because as mentioned before, if you have, say, AMP regulating CAMP bound transcription factors, physiologically, some of these group behaviors might also be very physiological in nature. So they might be in competition or they might be synergistic to AMP signaling, depending on the situation. So here's a view of the gram-negative Lux genes, and Lux genes can be found in a great number of different bacteria. And we're going to see this week, this concept of bioluminescence through the expression of something called luciferin and luciferase. So luciferase is the enzyme, and luciferin is oxidized by luciferase, and when luciferin is oxidized by luciferase, it produces photogenic light, not fluorescent light, which requires excitation for expression. And this is expressed in a cell dependent manner. So the more cells you have, or I should say, the more signal you have, even in the lack or the absence of number of cells, you get gene expression in a certain level of expression based on the amount of signaling protein you have. And the cool thing is that um, the more Lux protein you bind, the more Lux genes you transcribe and translate. So most bacteria with gram-positives or gram-negatives express another type of autoinducer called autoinducer 2. Autoinducer 2 is considered the universal autoinducer because it typically has the ability to bypass the peptidoglycan layer and the membrane as a or bind to two component regulators, but it is found, unlike AI1, which is signal peptides and acetylcholine lactones, AI2 has the ability or has sequence homology to bind to receptors uh, both inside and outside of the cells in gram positive and gram negative. So because it's a cross species and a cross uh, gram type regulatory cassette, it's considered the universal factor. But it works basically the same way that acetylhomoserine lactones and signal peptides do. It's just that it's done with a singular autoinducer that's not different between the two gram types. And then finally we have autoinducer 3. Autoinducer 3 is almost predominantly a two-component regulatory autoinducing molecule. And again, they call it an autoinducer because it induces more expression of itself, but also because the cells that express it can also receive it. So if I'm a cell and I express this molecule, there's nothing preventing my cell from binding the same molecule that I bound to. AI3 has a really interesting kind of history behind it in that AI3 looks and it is a charged and polar molecule, in that it also has the complexity, and all of these autoinducers have increases in the level of complexity and therefore the increase in complexity of the receptors they bind. And the increase in complexity in this case is that it looks very much chemically like epinephrine and norepinephrine. So now we're talking about autoinducers that look more like other types of signaling molecules that are more conserved in other organisms. So the evolution of these autoinducers went from very simple, uncharged, nonpolar to having a distinctly larger complexity in their molecular structure and their polarity, and they bind to similar receptor domains as you would find across different species and even genuses across different kingdoms. So obviously, higher order eukaryotes respond to epinephrine and norepinephrine, and they're two of the most predominant types of um, neurotransmitters in the body because they're involved in the adrenal response, and the fact that bacteria can respond to them and that they can produce a molecule that can express and change or probably bind to the same receptors that bind epinephrine and norepinephrine suggests the kind of beginning of the evolution of not only how bacteria can respond to the host and the host respond to the bacteria, so that the possible crosstalk between not only systems in the bacteria but also in between different species is really important. So quorum sensing itself is really important in regulating a lot of both phenotypic and genotypic changes as far as transcription and translation are concerned. But one of the other really important signaling molecules that we see inside of um, prokaryotes is cyclic di-GMP. So this is the byproduct of the degradation of GTP. And as we start getting an increase in cyclic di-GMP or guanine monophosphate, it is another intracellular cue, just like cyclic AMP is, that has to be considered in relationship to all the other uh, effects going on in the cell. So this is another measure of energetics and physiological changes inside of the cell. And as such, it is an important kind of bitopic regulator of how cells can regulate genes via quorum sensing, as well as all the other environmental conditions that they might come across that might affect the ability to undergo certain kind of physiological changes and or stressors inside it or outside of the cell. So um, that does it for genetics. I hope you enjoyed, and let me know if you have any questions in class.